Okay. Now we are going to discuss about structure of nephron. Structure of nephron. So structure of nephron or uriniferous tubule of kidney. Okay, this is the fundamental unit of our kidney, because so the function of the kidney, it means the urine formation can takes place in these nephrons. Okay, before going to discuss about the urine formation, we want to discuss about the structure of the nephron or uriniferous tubules. Uriniferous tubules. We know very well our kidneys consist of two regions. One is cortex region and another one is a medulla. In this medulla region, we can observe pyramids. In this pyramid, we are observing a microscopic arrangements or microscopic structures. That microscopic structures are known as a nephrons. Nephrons antam. Okay, so in this nephron we can observe mainly two parts. One is a Malpighian body. Malpighian body. And second one is a renal tubule. These are the main parts present in the nephron or uriniferous tubule. So Malpighian body consists of mainly, it is divided or it is having two regions. One is a Bowman's capsule. And second one is a glomerulus. Are you understand? So mainly Malpighian body and renal tubular body. So here Malpighian body consists of mainly two regions, distinct regions, that are a Bowman's capsule and a glomerulus. Okay, so now we can observe the in renal tubule we can observe one is a PCT. It means proximal convoluted tubule. Second one is a loop of Henle. Third one is a DCT, distal convoluted tubule. Very important one. So this, along with this, we can observe collecting duct. So these are the regions present in nephron. So renal tubular region consists of mainly four portions. One is a proximal convoluted tubule. Second one is a loop of Henle. Third one is a DCT, distal convoluted tubule, and collecting duct. So in this manner, the blood is filters and it can form the urine. బాగా అర్థం చేసుకోవాలి. సో ఈ విధంగానే ఒక్కొక్క రీజియన్లో మనకు బ్లడ్ ఫిల్టరేషన్ అనేది జరిగిన తర్వాత మనకు యూరిన్ ఫార్మేషన్ అనేది ఎలా ఏర్పడుతుంది అనేది మనం యూరిన్ ఫార్మేషన్లో చూస్తాం. బట్ హియర్ వీ కెన్ అబ్జర్వ్ ఓన్లీ స్ట్రక్చర్. What are the parts present or regions present in the nephron? Are you understand up to here? Yes, sir. So next one, we can, if you see the structure of the nephron, please write it. So if you observe the structure of the nephron, first region we can observe a distinct blind cup-like shape. Which one? Cup-like shape. This cup-like shape is known as a. This cup-like shape or structure is known as a Bowman's capsules. Bowman's capsule. I understand? So in this Bowman's capsule, a arteriole or artery, renal artery comes as a efferent arteriole. Efferent efferent arteriole, okay, and it can form a blood capillary network. Blood capillary network. This blood capillary network is known as a. This blood capillary network is known as a glomerulus. Glomerulus. Understand? Yes, so now we can observe a distinct regions in the first region of nephron, that is a Bowman's capsule, and it have a glomerulus. These glomerulus formed by the artery or arteriole. Artery comes as a arteriole. It can form the blood capillary network or blood capillary network, and it can comes out as a efferent arteriole. Efferent arteriole. Okay. So the Bowman's capsule is having a blood capillary network in it, that is known as a glomerulus. Glomerulus. This blood capillary network or glomerulus formed by the efferent arteriole. Afferent arteriole. If you observe, it comes out as a efferent arteriole. Comes out as a efferent arteriole. Very important. The size of the efferent arteriole is bigger than that of efferent arteriole. This construction, this important character can have, the character can lead to filter the blood. Okay. So arteriole anedi, so arteriole carries blood, oxygenated blood to the nephrons or kidney. Okay. So if the size of the efferent arteriole is bigger than that of efferent arteriole, that is helpful for the filtration of blood in the Bowman's capsule. Why? Because it can give a pressure. It can give a pressure. If for example it is a big size in, okay, it is smaller size. Why? Because the blood flowing in it, so automatically it can give some pressure. Okay, so that pressure is helpful to the filtration of blood in the Bowman's capsule. Are you understand? Yes, sir. So now this is very important. We can discuss later about the urine formation. Okay, we can in detail we will discuss there. Next one is this region is known as a PCT, proximal convoluted tubule. So next one. This region, a hairpin-like structure, is known as a loop of Henle. Loop of Henle. Okay. Next one, the next part is known as a DCT, distal convoluted tubule. So PCT also known as a first convoluted tubule. This is known as a second convoluted tubule. So and this efferent arteriole can form a blood capillary network around the this efferent arteriole can form the blood capillary network around the tubular part of the renal tubule of the nephron. Okay, so ఇక్కడ మీరు బాగా గుర్తుపెట్టుకోవాలి. Efferent arteriole is forming a blood capillary network around the tubular form. It means PCT, loop of Henle and DCT. Okay, it is a very fine structure, very important one. Okay, so here we want to discuss another important thing. So we are discussing glomerulus is forming, or it is filtering the blood due to the presence of pressure of blood or flow of the blood in the arterioles. Okay, as well as the structure of the blood capillary also helps in here, because so the blood capillary it have a lining of cells. It is a single lining of the cells around the blood capillaries. Okay, so these cells is made up of this glomerulus or blood capillaries are made up with a lining of epithelial cells or squamous epithelial cells. These squamous epithelial cells is also known as a podocytes. This arrangement, this structure is helpful. These podocytes can filter the blood in a Bowman's capsule. Capsule, very important. Okay, so one is a maintenance of the pressure. It lead to filtration, as well as the structure of the glomerulus also helpful for the filtration of the, why? Because it have a podocyte cells. These podocyte cells, epithelial cells, can filter the blood and it can form the primary urine. Which one? Primary urine. Okay, this primary urine, what is the difference between the blood and primary urine? Okay, so the primary urine cannot have blood cells. Blood means blood composition, which is having all the materials can same as in primary urine, except blood cells తప్ప మిగతా అన్ని మన బ్లడ్ లో ఉన్న అన్ని మెటీరియల్స్ అంటే న్యూట్రియంట్స్ కావచ్చు, అలాగే వేస్ట్ మెటీరియల్స్ కావచ్చు, వాటర్ కావచ్చు, అన్నీ కూడా మనకు ప్రైమరీ యూరిన్ లో ఉంటాయి. అంటే బ్లడ్ కాంపోజిషన్ లో ఉన్నటువంటి అన్నీ కూడా ప్రైమరీ యూరిన్ లో వీ కెన్ అబ్జర్వ్. ఓకే, ఎక్సెప్ట్ blood cells. Okay, so podocyte cells are helping for the filtration of blood, and also the size of the efferent and efferent arteriole can maintain the pressure. That pressure also helpful for the filtration. Are you understand? This is the structure of the nephron. Okay, so these if the forming efferent arteriole is forming the blood capillary network around the renal tubule. This arrangement can helpful for the exchange of materials. It means urine formation also takes place in this manner. Okay.